Hello students, this is lesson 2 for the chapter Cell, the Unit of Life. In this video, you will get a basic idea about the different types of cells that makes the living world. So let's discuss about the overview of a cell. So let's start with the size and shape of the cell. So as you know that the cell, it differs in size, shape and its functions. The smallest size of a cell that can be around 0.3 micron meter in length, and that is found in case of mycoplasma. Therefore, we can say that the mycoplasma, which is a unicellular organism, so that bears or that is the smallest cell in the whole living world. While as we know that the bacteria, that those are also not visible to our naked eye. So we have to take the help of a microscope in order to observe a bacteria. So bacteria, they measures around 3 to 5 micron meter in length. Similarly, the longest cell, that is the nerve cell or the neurons in case of the human body, out of all the cells in the living world. Similarly, the largest cell, that is found in case of the egg cell of an ostrich, which is a bird. Similarly, in case of the human beings, as I have said, the longest cell, that is the nerve cell. Now, the human red blood cell, so that measures about 7 micron meter in diameter, and these nerve cells, they can reach up to 90 centimeter in its length. Similarly, in case of the plant kingdom, the fibers or the sclerenchymatous tissue that we know, so they measures around 30 to 90 centimeter in case of jute and over a meter in case of hemp. So this is the overall size of a cell in case of the whole living world. Now let's come to the shape. So in case of the shape also, what we see that differs. They may be disc-like, or they can be polygonal, columnar, cuboidal, amoeboidal, thread-like, or irregular. As for example, the RBC or the human RBC, they are biconcave in its shape. Similarly, the nerve cells, they are polygonal in one part, and the other part, that is like the thread. Similarly, in case of the muscle cells, as you can see in the diagram, so they are spindle shaped. Then comes the other plant cells, which are the fiber like, so they are also long fiber like, and they have the tapered ends. So in case of the cells in both the multicellular category or the unicellular category, as you can see in the diagram, that the paramecium, that is also a unicellular structure, they have a different type of cell. Similarly, in case of the bacteria, they also bears the different types of cells based on their shape, that they can, some of them can be comma shaped, some of them they are round in structure, some of them they are rod like in structure, etc. So these cells, they come in different shapes and sizes. The living organisms are classified based on two types of cells, and these two types of cells, that is distinguished on the basis of the presence and absence of the true nucleus. And these two broad categories of cells that exist in the living kingdom are the prokaryotic cells and the eukaryotic cells. Now, what are prokaryotic cells? When the cell possesses a true membrane-bound nucleus, then it is called as a eukaryotic cell. Here the meaning of eukaryotic, that means true nucleus. Here, if we separate these two words, EU, that means true, and karyotic, that has come from the other name of nucleus, which is also called as karyon. And when this true nucleus it is present, which is a membrane bound structure, then it is called as a eukaryotic cell. The nucleus, it bears the genetic material which is called as the DNA. Now, in case of some primitive cells, their genetic material, that is DNA, it is not bounded by a nuclear membrane, and hence their genetic material, that is spread over an area of a cell. Now, those primitive cells, as they do not have the membrane bound or the nuclear membrane that encloses the genetic material, such type of cells are called as the prokaryotic cells. So these two types of cells, it is found mainly in the living kingdom. The primitive cells, they are the prokaryotic cells, where they do not have a true nucleus, but they have the genetic material which is naked, and that particular region within the cell that possesses this DNA or the genetic material, that is called as the nucleoid. So though they do not have the membrane bound nucleus, but they have the nucleoid. And the eukaryotic cells, they have a true nucleus, and hence the word eukaryotic is used there. Now let's have some idea about the prokaryotic cells in details. Although the prokaryotic cells, they have many differences other than the presence and absence of a true membrane bound nucleus. So let's have a study of the general organization of a prokaryotic cell now. These prokaryotic cells, they are morphologically the most primitive cells here on this earth, and they seem to have appeared about 1.3 billion years ago. Therefore, the organisms that possesses these prokaryotic cells, they are mainly the bacteria, blue-green algae, mycoplasma, pleuropneumonia-like organism, which we shortly call it as the PPLO, or it can be Rickettsia, Chlamydia, etc. So these prokaryotic cells, they have the structural organization in the form of an outer envelope, and therefore they are regarded as the single membrane or one envelope system organisms. Here, if we discuss about the structure of a prokaryotic cell, then at first comes the size and shape of a prokaryotic cell. They are generally smaller, but they multiply more rapidly than that of the eukaryotic cells. And they have varied structures, such as that of the coccus type, which is a round-shaped structure. Round-shaped structure. Then bacillus, which is a rod-shaped structure. Then comes spirillum, which is a spiral-shaped structure. Similarly, there are some coma-shaped structures. Now let's discuss about the outer envelope of these prokaryotic cells. So generally, these prokaryotic cells, they have an outer covering. And this outer covering, it is covered by an outer slime layer or capsule, a middle cell wall, and the inner cell membrane. So they have this three-layered outer envelope, and together that is the cell envelope. Now, what is slime layer? On the outer side of the cell wall. So basically we know that all the cells, they in case of basically the plant cells and the fungal cells, they have a cell wall. Now outside the cell wall, in some case or in some types of bacteria, and that is basically the gram-negative bacteria, they have an outer coating of loose gelatinous mass, which is called as the slime layer. And when it becomes thick and tough covering, then it is called as the capsule. Now, what is this gram-positive and the gram-negative bacteria? This gram-positive and gram-negative bacteria, they were named after a stain which is called as the Gram stain, which was made by or invented by Christian Gram in order to observe the bacteria in the microscope. And this gram-positive bacteria, they can retain this crystal violet colored stain, and those type of bacteria, that is called as gram-positive bacteria, while those which cannot retain the stain and they lose the stain after some time and we cannot observe them under the microscope, then it is called as the gram-negative bacteria. Now in case of this gram-negative bacteria, they have this slime layers, which is a loose gelatinous sheet that comes outside the cell wall. Now in case of them, in case of the prokaryotic cells, the cell wall, that is made up of material which is called as peptidoglycan. So that is the second envelope or second wall that the prokaryotic cells have. And some of the prokaryotic cells, such as mycoplasma, they do not have the cell wall. And then comes the cell membrane. So this cell membrane, this is a semi-permeable wall through or which controls the movement of molecules into and outside the cell. The cell wall or the cell membrane, both are responsible to protect the cell, also to maintain its shape, provide support and strength to it. Then next, let's come to the inner part of a prokaryotic cell. And this inner part of a prokaryotic cell, let's start it with the mesosome. Now mesosome, this is a structure which is we can call the infolding of the cell membrane. As you can see in the diagram, that the cell membrane that gets folded or that grows towards the inner side, and that becomes a bag-like structure or an extension. So therefore it is said that the special membranous structure is the mesosome, which is formed by the extensions of the plasma membrane into the prokaryotic cell. And this structure, it is in the form of vesicles, which is the round shaped structure, or it can be in the form of tubules, which can be a tube-like structure, or it can be in the lamellar form. Lamellar form, that means they can occur as some layered sheets. Now, what is the function of this mesosome? So mesosome, they help in the cell wall formation, then DNA replication, then distribution of the daughter cells, and they also plays a very important role in the respiratory process or cellular respiration, secretion, or to increase the surface area of the plasma membrane and the enzymatic content. Now let's concentrate on the cellular respiration part. As you know that in case of respiration, the respiratory substrates, they break down in order to form energy. Say for example, in case of our cells, the glucose, it breaks down and it forms the energy which is called as ATP, which normally in case of the eukaryotic cell, that takes place in the mitochondria. Now the same process that takes place here in the mesosome, where they stores this enzymatic or respiratory enzymes, which helps them additionally in the cellular respiration process in order to attain or gain the energy. And in some prokaryotes like cyanobacteria, there are some other membranous extensions in the cytoplasm which are called as the chromatophores, and this contains pigment. Say for example, in case of cyanobacteria or the photosynthetic bacteria, so they have this photosynthetic lamella or the thylakoids, which stores the photosynthetic pigments, and these are also derived from the cell membrane, which are present inside the cytoplasm of these prokaryotic cells. Then the prokaryotic cell, that bears a very important structure which is called as the ribosome. Ribosome is the only cytoplasmic organelle found in all the prokaryotic cells. So in prokaryotes, these ribosomes, they are associated with the plasma membrane of the cell, or they can remain freely in the cytoplasm. And they are about 15 nanometer by 20 nanometer in size, and it is mainly made up of two subunits. And these two subunits, that is called as 50S and 30S units. And together, this 50S and a 30S unit, here the 50S, that is the larger subunit, and the 30S, that is the smaller subunit. So these two subunits, they together, they make a structure which is called as the 70S, and in case of the prokaryotic cell, that is called as the 70S ribosome. More details of this ribosome that will be studied while discussing the eukaryotic cell. Now these ribosomes, in case of either the prokaryotes or the eukaryotes, they have the similar function, and the function is that the ribosomes, they are the sites of protein synthesis. These ribosomes, they may remain attached with a single RNA, which is a nucleic acid, and they forms polyribosome or polysomes. In simple words, we can say that when the ribosomes, they occurs in rows, then that is called as polyribosomes or polysomes. Now along with that, the prokaryotic cells, they also have some inclusion bodies which can store the reserved food material in case of a prokaryotic cells, and therefore they are regarded as the inclusion bodies. And these the inclusion bodies that can be in the form of gas vacuoles. So these gas vacuoles, they are free floating forms, and they can also be photosynthetic in nature, and they are permeable to atmospheric gases, which keeps the bacteria floating on or near the surface of water, or receive light or for photosynthesis, or to avoid strong lights. They are not bounded by any kind of membrane. As you know that the prokaryotic cell, they do not have an inner compartment system or inner envelope system. Therefore, we call it as the one envelope system. and therefore they remains in the form of phosphate granules, synophysine granules, glycogen granules, etc. Now let's come to the outer part of the prokaryotic cell or a bacterial cell that we are mainly referring to. So here, in case of the bacterial cell, they have external appendages. So these appendages helps them for movement or some other kind of activity. So then the bacterial cells, therefore, they can be motile as well as some can be non-motile based on the presence and absence of these appendages. The bacterial cell, they bear flagella. So this flagella, this is a long structure, fiber or hair-like structure, which is formed from the protein which is called as flagellin. And they act as a filamentous extension from the inner part towards the outer part, and this external part that is called as the flagella. So a bacterial flagellum that is mainly composed of these three parts. The one is the filament, then the other part that is the hook, and the lower part which is towards the inner part of the cell, so that is the basal body. And out of this, the filament that is the longest portion and extends from the cell surface to the outside. And this flagella, they helps in the movement or locomotion of a bacterial cell. Now along with the flagella, there are some other structures which is called as the pili or the fimbriae. Now what are pili? So pili, they are short, tubular, non-motile processes or appendages that is present on the surface of a bacterial body, and they are mainly responsible for the attachment to surfaces or to food or to any other organism, and they also help or take the help of this pili to attach itself to another bacteria for the purpose of conjugation, which we can call them to be the sexual reproduction. So therefore they also access the sex organ, and in that terms that is called the sex pili. And one such bacteria, that is the Escherichia coli. Next, this fimbriae, they are also same small bristle like fibers that are found outside the bacterial cell, and they also does the same function, that is for attachment to surfaces, to food or to other organisms. So this type of appendages, it is found in case of the bacteria, but the other organisms such as cyanobacteria or the blue-green algae, mycoplasma, they do not have such kind of appendages, or which are generally the flagella, pili and the fimbriae. So this is specific to bacteria. Now let's discuss about the nucleus now, which is present inside a bacterial cell. As you know that the prokaryotic cell, they do not have a well-defined nucleus. So it is said not well-defined because they do not bear the nuclear membrane which encloses the DNA, that is the genetic material within the cell. So therefore here the genetic material, that is naked, and it is confined to a particular region of the cytoplasm, or which is suspended in the cytoplasm in a particular confined region. Now that particular region, this is called as the nucleoid. This nucleoid, they are the compactly coiled DNA, which is the genetic material for the bacteria, that is present, and it is surrounded by the cytoplasm from all the sides. Here the DNA that is present in a compactly coiled structure, and as you know that particular region, that is only called as nucleoid. Now let's come to the structure of the DNA. So DNA, that is a double-stranded helical structure. It is a long double-stranded helical structure. And this helical structure that forms this compactly coiled nucleoid within the prokaryotic cell. And this is called as the prochromosome, or another term that is given here, this is called as the genophore. Just because to distinguish it from the eukaryotic chromosomes, as you know that we have chromosomes in our cells. Say for example, human beings, they have 46 chromosomes in a diploid cell. And here, in case of a prokaryotic cell, they do not bear a chromosome structure, but as they are compactly coiled and they resemble like that of a chromosome. So, in order to distinguish between a chromosome and the nucleoid, here a term it is given in case of the prokaryotic cell, which is called as the prochromosome or as the genophore. Now, in case of a prokaryotic cell, the DNA that also comes in the circular structure. They are also present in circular structure, which is called as the plasmid, which is the specific character for the prokaryotic cells. So these plasmids, they are the circular DNA molecules that is found in case of a bacterial cell, and mainly here the plasmids, they take some special functions, such as it helps them to have resistance against the antibiotics, or it helps them to have resistance from the attack of some other DNA, such as the viral DNA, which we can regard it as the foreign DNA. So in this way, the nuclear structures or the genetic material of the bacteria or the prokaryotic cells, they plays a very important role for the study purpose of many research works that is taking place in biotechnology or bioinformatics, etc. So in today's class, we just discussed about the structure of a prokaryotic cell in reference to a bacterial cell. I hope you have understood the content, and the points that were discussed today were completely based on the NCERT textbook. So I suggest you to go through the NCERT textbook. Thank you.